Um, Good morning, everyone. Thanks for having me. The name of my talk is Deep Amortisation for Rare Cell Discovery. So our work concerns single-cell observations. We care about discovering rare cell types from the data. These can lead us to a better understanding of disease or even earlier disease detection, especially in something like cancer. But rare groups are, as you can imagine, difficult to detect. There's this trade-off between precision and sensitivity. And on top of this, in this kind of data, there's actually no underlying known ground truth. So we currently focus specifically on single-cell RNA-seq datasets, and RNA-seq datasets contain observations of like 20,000 genes and thousands to now like hundreds of thousands of cells. And many cells just simply do not express certain genes, so these present as zeros in the data. And a typical analysis pipeline involves selecting the top highly variable genes, which is basically a measure of gene expression and variance. And then PCA is performed, so principal component analysis, where the cell observations are projected onto the principal components. And then distances are found between these projected cell observations using Euclidean distance, from which a k-nearest neighbor graph is built, on which the clustering algorithms are used. And then annotation is done based on these resultant clusters to try and identify like what these clusters are in terms of cell types. Okay, so this slide is basically to show you that pre-processing affects detected clusters. This plot is showing how the clusters and different approaches are related. You can see on the left, this is just like the standard approach to clustering. And next to it is the same thing, but without using any PCA. So you can see that there is a difference in identified clusters, and particularly the rare clusters that are identified, which you can also see along the bottom on the UMAP projections. And then next to this, I've also included something called highly deviant genes, which have been recently proposed in literature as an alternative to highly variable ones. And again, we see these differences in identified rare cell types. So then the question is, like, how do we actually interpret these choices? So pre-processing affects clustering results, but it also affects clustering interpretability. And that pipeline that I talked about PCA projection and then using Euclidean distance, basically it becomes very unclear what genes and what gene properties are driving clusters. And the way that we actually think about cell identities is linked to genes themselves. Biologically, we know that cell types are often defined by certain things being on and off. So we hypothesize that binarizing the data might help with both feature selection and also clustering. More uniform features, and in the limit, binarized features, will lead to more interpretable clusters as the contribution of each gene to cell distances becomes comparable. If we have a greater understanding about what genes are causing rare clustering, then we can make a more formed decision about like whether that cluster is even relevant. Okay, so the next task is basically how do we define a binarization for each gene? If we look at this plot on the right, this is kind of like a representative histogram of gene expression for a certain gene. And you can see what I was talking about before, where you have a lot of zero reads. And then you have this distribution of expression, which we're calling like the on distribution. And ideally, we want a model that can identify the zeros, but also maybe some background expression and map those to off, and then map that kind of on distribution. So one option is Bayesian inference under a Gaussian mixture model with a Dirichlet distribution, which is just a fancy way of saying a mixture model with a varying number of components to map that, map to the zeros, map to the off background expression if it's there, and map to the on. And we found in practice that using something like this has a few issues. So instead we opted for something called amortized inference, which in this context refers to making an optimization problem a prediction problem. So basically you train a model with a bunch of simulated data, and you get that model to predict the underlying parameters of the simulated distributions, and also the classification of on and off. And then once that model has been trained, you can just use it for prediction on real data sets. So in this case, we used a deep set transformer network to do this, and we trained it on distributions assuming this kind of Bayesian gas and mixture model that I mentioned before. But the cool thing about this is that it's distributionally flexible. So it's really easy to include certain priors through the data. In this example, train it on data that kind of looks like RNA-seq data. So this is kind of just like a diagrammatic overview of what I talked through. You have these training distributions with one or two components with like varying means and variances, and also these kind of peaks at zero that we see in the real data. And then once the network is trained, we use it to binarize each gene independently. And then from this, we do feature selection, and we select the 1,000 top bimodal genes, which means those genes where approximately half of them are mapped to this on state and half of them are mapped to this off state. We then perform clustering as normal, but also using this, the fully binarized profiles using a binary distance metric. And that's because we wanted to investigate the effect of both binarization and feature selection. So this is just a comparison of the different features that I talked about. Along the top, we have the highly variable genes, in the middle, the most deviant, and on the bottom, the most bimodal. And you can kind of see visually that these look more uniform. The plot on the right is just a quantification of this. It's a statistical test that measures the similarity in the gene distributions between each of the chosen feature sets. And you can see that indeed, the most bimodal genes are the most similar, apart from this kind of subset of highly variable genes, which when you dig into it, it's actually selecting genes where nearly all of the cells are mapped to zero, and a few of them have these kind of high expression values. And it's unclear how useful that would be for clustering anyway. Okay, so these are some of the clustering results. Again, you can see that, you know, a different number of rare clusters are found. But I think the interesting thing is that there are certain clusters that are consistent throughout the methods. And this is interesting because there's actually not much overlap between the most bimodal and the most highly variable selected genes, and yet they're finding the same clusters, which indicates that cell identities, you know, are defined by many genes. So then we wanted to investigate the properties of the resulting clusters and what that might tell us about the approach. So on the left here, we are looking at the differential expression between each of the clusters. And you can see that when you remove the gene set that's used for clustering, that actually the binarized and the bimodal approach have more differentially expressed genes between the clusters. So this indicates that it's almost generalizing better away from what you're clustering on. We also performed a binomial test to test how clusters split genes by their on and off interpretation, because we know that differential expression is going to favor those genes that have a higher average expression and higher variance around that. But that's not necessarily more biologically meaningful. And even when we look at genes not included in the clustering gene set, we again see that both the binarized and the bimodal approach are doing that more. And finally, we just looked at the correlation between cells within each of the identified clusters, and we found that the binarization approach especially is resulting in more similar clusters. So together, these results are suggesting that clustering on bimodal genes and further binarized genes are resulting in clusters that generalize better away from those genes that you've used to cluster them on. So just to conclude, we've used a deep amortized inference network to basically binarize expression data in a more informed way. We can select bimodal features and use binarized data for clusters to give a more interpretable and seemingly more accurate results, which is important for rare clusters. With our trained network, switching from the most variable to the most bimodal genes in a standard pipeline is pretty quick and easy. And with that, that's the end. Thank you for listening, and thanks to my supervisor, Chris Yao.
All right, thank you, Ellen. Do we have any questions for Ellen? So I have one question. For your dark network training, you shared a distribution of, I think, 9 different distributions. I wonder whether you've optimized a number of different types of distributions, whether that has an effect.
Yeah, so the way the data is simulated is that like we're basically training it to do Bayesian inference like under a Gaussian mixture model, but like each of it's trained on like a bunch of different distributions, all of which have a different mean and have a different variance. But yeah, it's like pretty easy to change those distributions to like more accurately reflect whatever you're doing.
Thank you. Question?
Hi, Michelle from the Peter MacCallum Cancer Centre. Very cool stuff. I was wondering if you've ever thought about what the effect of the data set on that approach could be. So for instance, if you have a data set with a lot of trajectories in development, do you think the binarization would still work?
Yeah, that's an interesting question, and like certainly where we want to take this work is basically like ensuring it's robust across different data sets and what that means. I mean, obviously transiently expressed cells are often quite rare, so it would be interesting to see what comes out using that. Yeah, thanks for the question.
And one last question.
Okay, thank you so much. That was quite interesting talk. When you are developing a machine learning model, model calibration and model generalization is quite important to be applicable on different data sets. So what type of strategy did you choose to make your model robust against variation observed in sequencing technology, sample preparation, or experimental condition to consistently identify the rare cell type?
Yeah, that's a good question. Basically, I guess like what we've done in our approach is like the same, like the point of it is that you're just basically doing prediction under like an assumed probabilistic model, which is the same as what you would do under any other model. So like, I think in this case, less so than other machine learning where you're training on really biased data. We're not training on biased data, we're training on simulated data under a probabilistic model. So I think it's like there's like not the same issues there. But yeah, like that is a good point. And I think one that we should definitely consider going forward is like how this generalizes. Thank you.
Thank you,